Hello students, welcome to the next session of DNA replication. I hope you are very clear with the semi-conservative nature of DNA replication. That means whenever DNA replicates, the two new duplexes will have one parental strand each and one newly synthesized strand. But it is not as simple as we see here. It just does not separate off like that and new strands are added up. No, it's a very complex process which involves several proteins and enzymes. So, how does this replication occur? Of course, they will be semi-conservative finally. The new duplexes will be semi-conserved nature, will have semi-conserved nature. But how does the whole process of replication occur? For that, first we need to understand certain terms like origin of replication, replication fork, replication bubble, role of deoxyribonucleotides, and what are those enzymes needed or involved in the replication process. So let us take it one by one, open up the whole mechanism. We'll follow up this video with the process. Here we will just understand the terms. Origin of replication. It's a specific nucleotide sequences where replication starts. See, this is a prokaryotic chromosome and this is a stretch of eukaryotic chromosome, DNA. So, in this circular chromosome, it cannot start replication anywhere. It should begin at a specific spot, at a fixed spot, referred to as ORI in short, origin of replication. That is where it starts replication. And for that, it has to have a sequence which guides that replication process. That sequences are bases, normally ATGC bases in a specific manner which initiates the process of replication. So when we compare the two types of cells, prokaryotic or eukaryotic cell, prokaryotes have just one ORI, whereas a eukaryotic cell has multiple ORI. You know why? Reasons are size. See the size of E.COLI, for example, prokaryotic cell, 4.6 million base pairs, and that too in a single circular chromosome. Whereas, you take the example of human, a eukaryotic cell, see the diploid content, it is 6.6 into 10 raised to the power 9 base pairs. And that is spread across or segregated into 23 pairs of linear chromosome, quite complex. So definitely one ORI cannot solve the problem of replication in eukaryotic cell, whereas it can manage it very well in a prokaryotic cell. So that is one reason. Secondly, as I said, genetic information on E.COLI is just on one chromosome. Of course, first reason is size. Second is only on one chromosome the information is there, whereas in eukaryotic cells such as human, it is there on 23 pairs. The DNA is there on 23 pairs of chromosomes and all those have to be replicated. So when you see this kind of complexity in eukaryotes, they have to have multiple origin of replication. So in human, this is just a short fragment shown here. Around 30,000 origin of replications would be there, spread across different zones, different regions of the DNA. So each of this ORI will be initiating replication simultaneously so as to finish up the whole process of replication in a normal rate, in a good specified time. Otherwise, it's just not possible to open up the DNA at one end and carry out replication throughout till it goes to the other end. 2.2 meters of DNA have to be replicated. So that's just not possible because it's a high energy needed process and that much of energy cannot be made available to open up the stretch from one end to the other end. So it happens in such short segments. Let's talk about the rate of polymerization. In prokaryotes, 1000 to 2000 base pairs per second have to be added. That means nucleotides one by one to be added. The enzyme DNA polymerase goes on adding nucleotides. That means this is the enzyme which carries out replication process. We will talk about the enzymes involved later. So the rate of replication, that means the rate at which that polymerase should add, is 2000 base pairs as given in NCERT. It is 2000. It ranges from 1000 to 2000 base pairs, but you read according to NCERT. And to cover up this much of nucleotides, or what we call normally base pairs, to be added, it should be done quite fast, because replication time of prokaryotic cell is DNA is given as 38 minutes. So approximately if we say 40 minutes, so this should be the rate of polymerization. Only then it can finish up each round of replication at that time, at that specified time. Now, you know very well that in eukaryotic cell the DNA replication and cell division must take place in a very highly coordinated manner. That means replication should be completed during the S phase, and only then mitosis starts. That means cell division phase starts after DNA has replicated. Please note one thing, that we are talking about replication of DNA in E. coli, that is mentioned as 38 minutes in your book, approximately 40 minutes suppose. So how can E. coli divide faster? Like in Messelson and Stahl experiment, we have learned that E. coli cells divide every 20 minutes. So how can E. coli cell divide faster than the time it takes to replicate? Replication time is 38 minutes. So here one thing should be very clear. Don't get confused with this timing. In prokaryote, there is no such restricted phase as S phase as we see in the eukaryotes for replication. So in a bacteria like E. coli, which is capable of very rapid growth in rich culture medium, with doubling time very short, that is 20 minutes, the replication time is much longer, 40 minutes. So what happens is, when a cell divides rapidly, suppose this is the ORI region, see how the replication goes on. From that ORI region, replication is initiated and it goes and terminates at one point. So it is bidirectional. And the new strand, see, you can see the red dotted strand is the newly synthesized strand for the blue shown strand. And here the blue dots are the newly synthesized strand for the original parental red strand. So this goes on. At an ORI it started and it goes on. But before one replication is completed, there is another ORI in this new one again starting replication. So what I mean to say is, within a specified time, there is what you call overlapping replications going on before the cell divides. So these are the ORI for future replication as the first round of replication is going on. So the what you call the separated strands with the newly synthesized strand also has an ORI, which is called future ORI. Both the strands have the same kind of ORI, which can also initiate replication before the first one is completed and even before the cell division is over. So that way, when you count, the replication time has to be counted before the cell division starts till the cell division is over. So it goes to approximately 38 to 40 minutes. And we have also discussed in genetic engineering chapters that ORI also controls the copy number. That means one cell can have more than one plasmid, depending on the copy number, how many replication rounds are over per cell division time. So that is all there in rapidly dividing cells. Next, let us talk about what is replication fork and replication bubble. See, where origin of replication starts initiating the process of replication, enzyme called helicase, again I'm not going to the detail of that now, cuts the hydrogen bonds at that area where ORI is there. Hydrogen bonds are disrupted and the strands separate, giving a Y-shaped appearance. So that Y-shaped here it is still wider, more opened. That means helicase is going on cutting the bonds both ways, that bidirectional it will open up. So you can see this Y-shaped area as a fork. You can see the two prongs like that of a fork. So it is called a replication fork. That is marked here in one area. Here, replication fork. So each of this is a replication fork. So when ORI starts replication at both directions, because helicase is opening the two strands or separating out the two strands by disrupting hydrogen bonds, definitely at one segment two forks are formed. So such two forks together make a bubble called replication bubble. So eukaryotes have many ORI, so many replication bubble. I have just circled one here. Each of this is a replication bubble. As many ORI as that many replication bubble. Whereas prokaryotes have only one replication bubble. At an ORI area, the two strands separated, so that is a bubble. So this also forms a replication fork. If you see this Y-shaped structure, it is a fork. So enzyme polymerase will go on adding nucleotides in those opened areas. One by one at a time, nucleotides will be added in both directions. Polymerases will start acting on both directions in eukaryotic cell, such that each of the bubble goes on becoming wider and wider. The fork opens up much deeper, cutting off all the bonds, and every area or every segment, when polymerization is going on by the enzyme polymerase, definitely they'll go and meet. Bubbles will widen and the whole segment would be opened up completely by the time replication is simultaneously completed in all these bubbles. Thousands and thousands of bubbles. So the original parental strand will separate up, having the newly synthesized strand with it. Similarly, the other parental strand also separates out with the newly synthesized strands incorporated there. So new identical daughter DNA duplexes are formed, which are exact copy of the parental duplex. Also note, the rate of polymerization in eukaryotes is quite slow compared to the prokaryotes. See, it's only 100 nucleotides per second. So with thousands and thousands of ORI, polymerase will start adding up nucleotides at this rate simultaneously in all these replication forks, both ways. Only then it can carry out the process of replication efficiently, because as such it is quite a complex process. Many things have to be taken care of. So if this happens from one end to the other end of a 2.2 meter DNA, it would take ages to finish up one round of replication cycle. Moreover, the new segments formed, of course, will be in pieces. They won't be joined, no. So enzyme ligase will take care of sealing them as one complete segment, one complete strand. So those gaps would be filled up. So here you can just see a detail of what is replication fork. We have already discussed it. Enzyme helicase will cut hydrogen bonds, so it is also called unzipping, creating a fork. And polymerase will start. We'll go to the detail of how polymerization works in the next video. So two replication forks make a bubble, just a recapping of all that. We shall talk about leading, lagging strand and all in the next video. Another important thing you should know is replication is carried out by abundant energy expenditure. That means it is not passive and spontaneous. Polymerization requires energy that comes from the breakdown of triphosphates of the deoxyribonucleotides. That is why it cannot be opened up at a stretch and new nucleotides can be added. It has to open up segment by segment like this, because it requires energy, and it is provided by deoxyriboadenosine triphosphate, deoxyriboguanosine triphosphate, deoxycytidine triphosphate, and deoxythymidine triphosphate. They serve as substrates. See, you can see them being added up by the polymerase. So they are the substrates on which the new chain grows on, and they also provide energy for polymerization. How do they provide energy? The terminal two phosphates provide energy, because this is the deoxyribonucleotide triphosphate. When the bonds between the phosphate breaks, phosphodiester bond is formed between the newly coming nucleotide and the 3' OH group of the growing chain. Okay students, please watch the next video for the whole process.